Development Program on emerging topics in computing, quantum, microfluidics, and memristors. Here, we are going to start the microfluidics track, session 1. We are going to have Dr. Sudeep Roy, IIT Roorkee. I am requesting our Honorable VC Sir to say few words for our speaker. Sir?
So Shudip, hello, how are you? Shudip, you're muted. Please unmute yourself.
Yes. Hello, sir. Yes, I am fine. How are you, sir?
I am fine. So actually, I have known Dr. Shudip Rai for many years. So he was
Nice meeting you,
from
sir.
IIT Kharagpur only. He did his PhD from there. And now he is a faculty member at IIT Roorkee. So means I had interacted with him since long while he had started his work on microfluidic biochips. Now it's really nice that he will be delivering a talk today. So we are all eager to hear from him. So what are the new development and the new things that has been carried out? So I think everyone is just waiting to hear from you, not from me.
No problem, sir. Thank you, sir. Nice talking to you. Nice meeting
So
after
Dr.
a long
Banerjee,
time.
please continue.
Thank you, sir. I am once again going to share my screen. Dr. Sudeep Roy is an assistant professor in the Department of Computer Science and Engineering of Indian Institute of Technology, IIT Roorkee, India since July 2014. He is also an associated faculty member of the Center of Excellence in Disaster Mitigation and Management in IIT Roorkee since April 2015. Currently, he is a JSPS Fellow in the College of Information Science and Engineering, Ritsumeikan University, Biwasco Kusatsu campus, Japan, during April 2021 to January 2022. He received his BSc degree honors in physics and BTech degree in computer science and engineering from the University of Calcutta, India in 2001 and 2004, respectively, and MS by research and PhD degrees in computer science and engineering from IIT Kharagpur, India in 2009 and 2014, respectively. He worked as research associate in the Department of Computer Science and Information Engineering at National Cheng Kung University, NCKU, Taiwan in 2014. He was a visiting research scholar in the Disaster Prevention Research Institute, DPRI, at Kyoto University, Japan in 2020. He has authored one book chapter, 2 granted US patents, including 25 research articles in international peer-reviewed journals and 40 papers in international peer-reviewed conference proceedings. His current research interests include computer-aided design for digital systems, optimization techniques, information and communication technologies for disaster risk reduction. He was a recipient of JSPS Invitational Fellowship Award for long term from the Japan Society for the Promotion of Science, Government of Japan in 2021, Early Career Research Award from Department of Science and Technology, Government of India in 2017, and Microsoft Research India PhD Fellowship Award in 2010. He is a member of IEEE and ACM. So today we are going to have his talk on microfluidic biochips and some computational problems for their design. Sir, this is now up to you. So please share your screen, sir.
Yes. Thank you, Tapolina, for your kind words and kind words to introduce me. Okay, so
Thank you,
let me
sir.
share. So the entire screen. Screen one. Okay, this is better.
So your voice is very low.
Oh, maybe problem. Hello. Now is it audible?
Yes, sir. Yes, sir.
Okay. Okay. So let me the slideshow mode. Yeah. Okay. Yeah. Just a moment. Yes. Okay. Yeah. So my screen is visible, right?
Yes, sir, it is visible.
Oh, okay. Yeah. Thank you, Topalina. Yeah. So, yes. Yeah. So, good morning, everyone. So, I'm going to talk about this microfluidic biochip and some computational problems for their design automation. And as you know already, 3 talks on this have been delivered by Dr. Tavolina and then Professor Ho and then also Professor Krish. So maybe some of the things may be common, but yes, I will also introduce some of the topics that I pursued during last few years. And yes, okay, so I will continue with this microfluidic biochip basics, and then okay. So before going to that, I should acknowledge my mentors, my professor, supervisor, Professor Bhargav Bhattacharya and Professor Parthapratim Chakraborty. They are now in IIT Kharagpur. And also I worked with Professor Krish during my PhD, and after that also with my collaborative research projects. So basically, I have they are my mentors, you can say. So Professor Krish also, I got motivated to this field by his work. He is the pioneer of the field, you can say. And then I have the current collaboration with Professor Shigeru Yamashita from Japan. I am currently sitting in his lab only. And Professor Sung-hee Ho, he is from Taiwan. You have already listened his talk. And Professor Indranil Sengupta, I also had a collaboration with Professor. And Professor Junder Huang, Professor Ulsisman from different universities, Professor Bingley, Dr. Bingley, Professor Hao, Hailong Yao, and others like Professor Sharmishta Ghoshal from IIST for bio-related work she does, and Professor Dr. Shukanto Bhattacharya from IIT Guwahati, Professor Devashish Mitra from NIT Durgapur, and also Dr. Sudeep Poddar from Austria. He is now postdoc in JKU University. And my students, I could not do the remaining work that I will discuss without the contribution made by the students. So I should acknowledge their contributions. So my PhD students, current PhD students, Debraj and Devashish, they are in IIT Roorkee, and my MTech students, so actually this Amiuddin is from IIT Kharagpur. He was a student, and we jointly worked with Professor Indranil Sengupta. And my other MTech students in IIT Kharagpur, namely the Satyendra, Varsha, Ananya, Shalu, and Nishant. And also there are a couple of BTech students who did very well, and they published some papers in IIT Roorkee. And I am very much fortunate that during my PhD, I was introduced with Professor Dr. Srijan Kumar. He is now doctorate, and he's a faculty now in Georgia Tech University. And he did his bachelor project with me in IIT Kharagpur with under the supervision of Professor P.P. Chakraborty. And also I acknowledge my internship student from IIST, Shohini Shah. Last year, she completed her BTech, and she is now going to Duke University for doing PhD, for her PhD. So the outline of the talk is basically, I will talk about a little bit of digital microfluidic biochip, the basics that we have, maybe some overlap with the other things, other talks. And the design automation problems, basically just some crisp view of the design automation problems. Already many discussion has been done by 3 different talks, but I'll also, in my own way, I'll discuss. And then some advanced microfluidic biochips, how they are looking, and how these different automation problems are arising for these advanced microfluidic biochips. I will discuss that. And then I will come to the automated sample preparation problem. That is not only just for digital microfluidic biochip, but there are many different types of microfluidic biochip which for which this automation is important, and people have studied this problem very well. Means there are a lot of papers on this. So I'll discuss about a few things on that, and our research recent results that we did during the during my collaboration after PhD and also with my students, and then I'll conclude. So basics of the laboratory on chip, as you know, that already you were introduced about this topic, that this is there are a lot of complex operations in the biochemical laboratories, and it requires technicians, skilled technicians, and also bulky equipment, so which basically incurs the cost. So that's why these kind of tests are basically taking is expensive, so we have to pay a lot for the test to be done. So that's why, and also since these are semi-manual, you can say manual or semi-manual, because sometimes some automated robotic hands are used, and along with the human being who are handling this these microfluidic operations, not microfluidic operations, you can say the biochemical operations in the biochemical laboratory or pathological laboratory. So these are basically time consuming because they are not automated. Many of the things many of the steps require manual intervention, and it's laborious, so it requires the skilled technician, and also it requires a considerable amount of fluids, so which we basically want to avoid, because as you have seen in the last lecture in the Professor Krish and Professor Ho's lecture, that the cost of the fluid is very high. So we have to reduce somehow this cost of the uses of the fluid. So whatever be the fluid, like sample or reagent, so you have to reduce the amount of uses. And if we can have this laboratory, the whole biochemical laboratory on a small chip, so which we can hold on our palm, in our palm, so then this can be automated. Because of this design, it requires the manual intervention. It is very difficult to do manually perform the operations, so it requires the automation, so it can be automated, the small chip. And since this is a very small dimension, so many of the operations, many of the modules, many of the functional tasks can be integrated, so as it is done in the VLSI chip. So lots of operations are being integrated, and that's why we are talking about very large scale integration. So in case of means IC electronics, we have seen the development and the history, basically evolution, how this technology has evolved during maybe last 20 years or last 30 years. So it has done a lot of technological advancement, and that's why we are nowadays we are holding so much smart devices in our hand. And so similarly, this microfluidic chip or microfluidic biochip, or you can say the laboratory on chip, LOC, are basically the goal is to make it automated, integrated, inexpensive, and disposable chip. So disposable because this is not like our smartphone. So we are dealing with the human physiological fluid. So as we are facing these infectious diseases, so we should not be very much happy to share the contaminated chip with others. So it should not be very good to do that. So that's why it should be disposable. So if we are talking about the cost and inexpensive and also disposable, that means we have to design the chip in such a way that the design and fabrication should not incur a lot of cost in it, so that we can sell it at very cheap price. So $1 one chip, so like that. Anyway, so basically in the biochemical laboratory, as you have seen in the biochemical laboratory, there are many experts or biochemists. They perform different operations manually. So the main objective is to make this whole laboratory into a small chip. So if we are going to do this, then there are two kinds of techniques that people have adopted. Like one is that they can deal with this continuous flow of the fluid. So that is basically channel based. So you can have different channels and on the chip, and then you can control the valves and pumps to perform some operations. And you can also deal with some another technology that has evolved, that is basically digital microfluidic biochip. We have seen that the droplets can be created from the continuous flow. So that is important, that how you can create some droplets and then perform the operations. So that will be easier for us to program it or reconfigure the modules. And yes, so that's basically the purpose of our means interest, basically why we are interested in from computer science and electronics. Why we are interested in microfluidic biochip, because we have seen this advancement that by programming we can do the chip design. So by writing the software, we can have this EDA tool, and basically that helps us to get the VLSI chip. So like that similar way, that discretization and also this continuous flow microfluidic base. So in that case, also this control of the valves, both are basically the purpose is to make it programmable and reconfigurable. So there are two different kinds of microfluidic biochip, as we have seen in the literature. So the first one is this digital microfluidic biochip. It's based on the electroweighting and dielectric. I'm not going into detail of that, already discussed. So this is there are a set of electrodes, and the fluid droplets can be manipulated with the help of voltages in the electrodes. And there are different versions of this kind of chip, like different dimension of the electrodes. There are different advancement people have done. The layout of the chip also not always regular rectangular shape. It may be of any polygonal shape, or it can have the hole inside the polygon. So like this different design, layout design has been explored, and it's basically based on the application. So most of the researches that you will find in the literature will be application specific. If you are talking about the actual microfluidic biochip design and fabrication and its use, so basically this application specific microfluidic biochip is the current target, as we see in the literature. And then also in the continuous flow microfluidics, we have these valves, and we can basically control the valves using microcontroller. So it's electronically controlled valve. So we can send the voltage signals to switch on and off. So it's just like 0 and 1. So you can just switch on and off some of the valves and make this mixing happen. So mixing of two different fluids, as you can see in this video. So these two videos are taken from two university video, real chip video. Basically, the first one, this digital one, is from University of Toronto, Wheeler Lab. They have this. They have developed a kit that is called Drop Boat, and it's a video from that. And this is from University of actually MIT. They have developed this kind of chip in 2009. They have published some paper, and they have this video in that paper. You can find there. So in the digital microfluidic biochip, I will not go too much detail of this slide, because already many of the things have been discussed in yesterday's lecture. So basics behind this, the basic physics behind this fluid droplet movement is basically electroweighting on dielectric. So if you apply the voltage, then the contact angle and the surface tension basically changes. And if the surface tension changes, the contact angle changes, then the fluid droplet start moving. And there are two different class of digital microfluidic biochip basically. One is this open end, means you have the bottom plate, which is actually array of electrodes, bottom array of electrodes, as you can see in this image schematic diagram. And you can also have a top electrode on top of this. So sometimes they talk about this confined digital microfluidic biochip. Sometimes they talk about this open one. So currently people are doing research on this confined one, because it is very difficult to move fix the droplet on the surface with the change of the gravity. So that's why it is making this confined one. So in the confined one, you can see there is an arrangement, basically LED and LED is LED. LED is the light that is sent through this electrode. And this ITO electrode is basically indium tin oxide. So indium tin oxide is basically a metallic behavior. It is conducting behavior. It can conduct the electricity, but it is transparent. So optically, if you send some light through this electrode, this will pass through the droplet, and it will go to the photodiode, which is there on the top electrode. So this so this photodiode will generate some voltage, and according to the calibration curve, you can imagine, you can calculate, basically you can estimate the concentration of this droplet, of a fluid, of a particular sample in this droplet. So yeah, this is actually this photoelectric effect that Professor Einstein has worked on. So this is a kind of module, you can say, where this LED and means photodiode is arranged with the digital microfluidic biochip. So this kind of arrangement is called basically detection unit, one kind of detection unit which is responsible for detecting the concentration. So concentration of the fluid. And there is another kind of detection unit also that is used for the volume measurement. So I will talk that later on. So these are all basics. So maybe I can skip. So we can have different fluid loaded into the reservoirs, and we can create the droplets. So that is called dispense, and then we can move them and mix them. Okay. So the first another thing is that this hydrophobic nature of the fluid. So if you make this surface hydrophobic, then you can make a droplet, spherical shape, near spherical shape droplet. So that's why this pyroline material, Teflon material is used. Pyroline is the dielectric medium, and Teflon is the your non-sticky Teflon you have used in the frying pan. So that's basically non-sticky hydrophobic layer. So that's the material aspect. So there are many different versions of means this technology. So rather in computer science, we basically try to understand this phenomenon, this working principle of the chip, and then model it as simple as a graph or in terms of some grid to make our algorithm ready. So okay, so the basic fluidic operations. So you have to understand the operations, what kind of fluidic operations we perform in the biochemical laboratory. So which we may also imitate in our in a microfluidic biochip. Oh, sorry. So this video is taken from that drop board video, which is Wheeler Lab, as I told you earlier. It's from University of Toronto. So they create this droplet that is called the dispense operation by sequentially. Basically, they apply the voltages to the nearby electrodes and suddenly switch off some of the electrodes. So it creates the droplet. And then you can move the droplet. So that is transport operation. And you can mix them, merge, means keeping them together, and then mix them by steering, by rotating. And then you can also split them. So you can see in the video, all these are happening. So these blue colors text, as I kept here, are basically the basic operations on the fluid. And you can also have some derived operations. So derived operations means that you want to detect some concentration of the fluid, of the sample in the fluid. So in that case, you have to move the droplet from the current location to the detection unit. So detection unit is not available on all under all the electrodes. So it is at a particular location. So you have to move there and then switch on the detection unit, like you apply the voltage and measure the current in the photodiode output, and then you tell about the concentration. So that's detection unit. So that's basically derived operation, because it is basically combining two different operations and then perform the particular operation. Like dilution, in that case also, you basically take two fluids and they are basically sample and buffer, and then you mix them and try to reduce the concentration. So that's basically the set of steps it may require, sequence of steps you may require in the dilution operation. That's why it is a derived operation. Like perfusion is also some kind of derived operation, and incubation or heating. So it is similar, as simple as that. Under some electrode, you have the heater. You may say that there are coil, basically heating coil, and you can apply the voltage. It will be heated, and that electrode will be heated, and that's why the droplet sitting on that electrode will receive that temperature, basically. So these kind of operations are basically moving and then switching on that particular device. Like cooler is also like that. So cooling of the electrode, the cooling arrangement is on, and then the droplet is moved to that particular electrode, and then you get this cooling done. So this kind of temperature controlling and all these type of operations are derived operations, which are composed of multiple basic operations, you can say. So depending on these operations, which are basically the task you want to do with the fluid, you can make different devices, or you can consider that you have to have different devices or modules that will perform that operation. So you can say that the mixer is the basic operation, like in a computer, we say the processor is the main operation that is doing. Then storage. So in computer also we have RAM or maybe hard disk. That is basically storage. So the storage unit is also required, because once you have prepared some fluid, you have mixed, maybe that mixed fluid will not be used in the next clock cycle or next time. There will be another operation that will be done in the mixer. So you have to store this fluid for maybe after 5 sequential steps you completed, and then you will require these mixers. So that's why storage is important. So you have to have some memory to keep it. So processor, memory, and then reservoir. So reservoir is the IO device basically. So reservoir is the input device. So you have the input port through which the fluid is loaded. So reservoir is a big size electrode on which there is basically through some capillary channel the fluid is loaded, and you can have the dispense operation by applying the voltages as EWD is doing. You can generate the droplets. So the reservoir is basically, you can see it as an input device, or maybe it can be output device also, because some reservoirs are waste reservoir. So some fluid which is wasted in the fluid not being used in the next clock cycles, those will be transported to that waste reservoir. So if that kind of waste reservoir is there in the periphery of the chip, then you can think that this is the output port. So this reservoir is basically mimicking that IO devices in our computer. And detection unit is basically sensing unit. So it's also, you can say that it is also basically input, or you can say the output device. You can measure the output voltage of the photodiode and tell about the concentration value. So it's basically cyber physical system. So that's why you are measuring the physical phenomena and telling that in terms of the computer cyber data. So like this, you can visualize the chip. So it is having processor, it is having memory, it is having the IO device. It also have the sensing and detection unit, which is cyber physical system. Okay. So as I was talking about, this is the arrangement of photodiode and LED based arrangement, which is basically used for measuring the concentration of the fluid in this droplet, and it basically follows the Einstein's photoelectricity phenomena and photoelectric effect. And the second one is very interesting here, is that you can imagine these two electrodes, like the bottom electrode and the top electrode, as a parallel plate capacitor. Parallel plate capacitor because you have the droplet in between, and there also you have the dielectric medium like pidyline and all this. But these two plates are metal electrodes, and you have some dielectric in between. So that's why it can be seen as a capacitive sensing circuit arrangement, where you can apply the simple physics that you have studied in your high school. That is, the capacitance C is equal to epsilon A by T, right? So epsilon is the permittivity of that medium, that is the dielectric medium, which is here in this fluid. It has particular epsilon value depending on the material that is being used. And the electrode are having some square shape, you can imagine, and the fluid droplet is sitting in between this. So this is the cross-sectional view you can see in this slide, but you have to imagine that on top view, that you have a square electrode, you have the fluid droplet, you have another top electrode. So this is a parallel plate capacitance, and you have the overlapping area because of the fluid. Fluid is of particular volume, so it will have overlapping area, that is A in your formula. So that is overlapping area with the electrodes. So parallel plate electrodes. So that area, if the volume of the fluid is changing, then that overlapping area will change. And finally, this distance between these two electrodes is basically D. So C equal to epsilon A by D. So if there is a change in A, then you will have a change in voltage also, because the capacitance will change. C equal to epsilon A by D. So that actually helps to measure the volume of the droplet. So when this volume of the droplet is measured, because many of these operations are not very perfect, so there are some imbalance happens. Like in the previous lecture also we have seen that the droplet can be splitted. We are assuming that they are equally. They are having the both equal volume, but it is not always the case. So you may have the improper, you can say that unbalanced splitting. So that there is a delta volume change. So delta change in the volume in both the. So if some is getting more, so that is plus delta, and here it is minus delta. So you can measure this volume change, whether it is within the tolerance limit. So there is some tolerance limit, because in this case, we all know, whoever designing the chip knows that there is a chance of this imperfection. So that's why there is a tolerance limit. Based on that, the protocol is defined, and it is being implemented on the chip. So, so these are the basically two different detection and sensing modules, which are very important in the digital microfluidic biochip, you can say. So now I will briefly go through this digital microfluidic biochips, or maybe you can say in general the microfluidic biochip design automation. So as we know that we have some, actually this can be seen as a software engineering task, because in real life, when somebody is telling you that I need this software which will perform these operations in this sequence, so the user or the customer is telling you that this is the sequence you have to follow, and these are the operations I have to do. So this sequence of operations is also there in biochemical laboratories. So they follow those sequence of operations, and that is basically called the real life. You can say these are the real life scenario. These are actually named as Bioassay or Bioprotocol. So both are same actually. So basically this protocol are important. So you are having some protocol to implement on the chip in the hardware. So now you are given the hardware. So let us assume that we are talking about digital microfluidic biochip here. So you are given a hardware. So hardware also not basically you have to design, as a designer, you have to design this hardware which will perform these operations. So what you need to do is basically first task is that whether you can precisely or unambiguously represent the bioprotocol or bioassay. So that is important task, that how we will represent that. So as we have seen in the software design, the software specification language. So basically people write the software requirements in different modeling language. So they have UML and all this. So here also we need some standardization of this protocol, because it cannot be directly read the manual of the biochemical laboratory, and it will be understood by us as a computer scientist and write the software and do it. So there is a gap basically in the research. So that's why the biochemical laboratories and these computer science people need to come together and sit to work on this. Okay, I will come to that with some examples also how they are working. And basically, so as an input, you are given this protocol in some format. So imagine that you have been given some file format, and you have to read this, and finally you have to implement that those steps, sequence of steps on the chip. And these chip you can see, as you can see in this schematic diagram. So it is having some reservoirs, so input reservoirs and waste reservoirs, as I told, that IO devices. And you have the processing unit like mixer unit. You have the storage unit like these brown electrodes, and you can see that dispensing unit, that is basically IO port. And you have these different mixing and splitting, different operations to do. So as in the processor, we have this adder, multiplier, and all these things. So like this, you have different modules, and you also have the detection unit in some particular electrodes. So you have to now map these whole protocol on this chip to perform. So that is basically the you can say the mapping task, and which is basically done through some sequence of steps, as we have seen in the VLSI chip design. So the digital VLSI chip design actually follows some sequence of phases. There are different problems, sub-problems that need to be solved, and then finally get this layout of the chip, that is GDS2 file in VLSI chip. So here also we'll have the layout of the chip, and also we need to know the control sequence. What will be the control? To which electrode, which voltage to means 0 or 1, high or low voltage to be applied at which clock cycle, at which timestamp. So that is important. So I was talking about the standardization of the language. So basically, there is a very good research that has been done by Microsoft Research India in very old maybe, but it has some other research also. I will discuss in the next slide, the recent one. And basically, they first talked about that programming language is important to understand and use to represent the bioprotocol and standardize that. So they use the C++ based basically. Okay, so the next slide will, yes, this one. So here you can see that they have used a similar syntax that is basically used in C++. So they have included hash include biocoder.h. So that is a header file. They define all the functions in that file. They can use this hash defined global variable to set some value there. And they are using this void main, all this syntax, and then start protocol to tell about the name of the protocol. The syntax, if you follow, it will look like that C++ or Java kind of programming syntax that is being used here. So the name of the protocol is some particular PCR test. We already know about this PCR test due to COVID-19. So this is a different type of PCR test. Actually, there are many different PCR tests are being performed in the biochemical laboratories to measure something. Okay, so basically the virus or maybe the microbacteria, they are very small in number, so they need to be basically increase the number. So polymer chain reaction helps them to integrate, means you can say that multiply their numbers, and then we can visualize, we can basically sense, we can detect that that one. Okay, so if you follow the syntax here, so fluid NaOH, so NaOH is kind of buffer solution, you can say here, is the amount, or you can say some mole, so that is basically volume of the fluid is taken as the input, and it is written as new underscore fluid. So it is a similar syntax that new we use that in the constructor, so to create an object. So it's similar to that. And what is the meaning of this? So when this line will be executed, that means the corresponding control sequence for the dispense operation of the fluid from the reservoir, input reservoir, will be executed. Okay, so that is basically the standardization. So this is a gap actually right now, what I feel is very important, and people are working towards this. So there are some, okay, so based on this bioprotocol standardization and description languages, I had a study on this, and then I found that in Nature Journal, you know already what is Nature Journal. So it's very top science journal, or you can say a very high impact journal. So they actually did a recent survey in 2016. And they found that they actually involved 1,576 researchers who states basically, so they did the survey, and then what is the survey basically? They took the experiments, those were already published in the Nature Journal, and then told the researchers to reproduce the same result after you do the same experiment again. So repetition of the same experiment, when these scientists or researchers were asked, then 70% of the researchers, they tried and failed to reproduce this another scientist experiments. So another scientist experiment, that means that the protocol is already written by the scientist, by scientist A, and scientist B wants to implement that, want to execute that protocol by reading that paper, not just directly contacting or just getting the information, anything, just reading that paper, want to execute that. Similarly, we face in the simulation problem, programming also in our work, that if we want to reproduce the same thing, so may not get the same result. So sometimes it happens, we also face this problem. So they actually surveyed on this, and they found that the protocol as it is written in the paper, in the research paper, is not complete, is not unambiguous. So there is some challenge basically need to address. So you can see the result here. The first one is still acceptable, that another scientist experiment is not repeated, and they are not able to reproduce the same result, 70% in such cases. But more than half of them fail to reproduce their own experiments. So even they have actually did this experiment in the biochemical laboratory, they have not written down properly that what was the environment, what was the steps precisely and unambiguously. And that was the main problem here. So this survey actually means basically tells about that where is the gap. So in the standardization and the bioprotocol description language, there is a lot of things is required to do where both biochemist people and the computer science people need to sit, because computer science people know the programming languages and all these details behind this, the principles of programming language and the compiler design they knows. So if they work together, then they can do something better. So based on programming language like languages like Haskell, C++, C, and okay, I have repeated Haskell again. Okay, so this Python and Go, all these languages, there you will find several notable attempts that has been done by different researchers, and these are published in very good journals, very top ranked journals. You can find this. I have not mentioned the citation here, but if you Google it, you can find all this. So the experiment actions, that is the full form. So actually exact is the keyword that they have developed. This is a standardization language, exact language. So they also come up with the exact 2, the second version of that language. Biocoder I already mentioned in the Microsoft research. Antha is another work, and Auto Protocol is also, nowadays people are basically using biochemical laboratory people. They are using this Auto Protocol and Bioprotocol, these 2 database kind of thing, where they are writing their bioprotocol in some standardized syntax. They're following some Auto Protocol is basically following the XML for syntax. So they follow that syntax to write down the steps, so that any other researcher can implement that protocol again. Okay, so this is a very important problem people are facing now. So that's why this microfluidic biochip, I feel that is getting some hindrance to progress further. It is getting delayed. So basically, if this research is why this is important, this is not only for microfluidic biochip, it is also important because if you see the year I have mentioned, 2008 to 2018, I found these papers, but still people are doing research. So these papers, if you look into, these are not only talking about microfluidic biochip, they are talking about the robotic arm-based system also. So in terms of the biochemical laboratories, which is having different advanced version of the robotic arm-based systems, they are also not able to figure out how to program that robotic arm so that these the previous protocol can be implemented again without any error. Okay, so that is basically one challenge I found. So now let us come to the simple life that we are dealing with in computer science. Basically, the sequence of operations that we tell about to be followed for the biochemical protocol to implement. So the simplest thing that we can imagine in computer science is that we write down the list of nodes and list of edges, because it is a directed acyclic graph. So you'll have set of nodes and set of directed edges. So these edges are basically telling the direction 1, 2, 3 like this, and the nodes you are basically having different attributes. So you will have the node ID, you will have the node type, the type of operations that you are going to do, and if there are multiple similar type of operation, then you have the operation identifier also, like dispensing 1, dispensing 2, like this. So also you are mentioning the volume of the fluid, so you can also write like this. So for the time being, if you follow this the sequencing graph, which is basically protocol, so it is telling that there are different blue nodes, which are basically the input of different fluids. You are dispensing the fluids in terms of digital microfluidic biochip, if you're thinking. So you have different dispense operations, and then you're doing the mixing operation in the yellow nodes. And after these yellow nodes are done, then you are going to the output node. So you're basically performing the sequence of operations as mentioned in this directed acyclic graph. And for these graph, you can write that particular text file to represent this, that these are the sequence of nodes, and these are the steps of, set of nodes, and these are the set of edges, and that's all. So you can read that file and then do your programming, do your automation. So that's to make this life easier. So people work like this in our domain, from computer science. So maybe this was already discussed in Professor Chris' lecture. I was listening yesterday also his lecture. So so maybe for simplicity, I just want to say one thing, that So given the protocol, that bioprotocol, we need to have the directed acyclic graph. So that is also called sometimes as sequencing graph model. So basically this graph is our input for our program. So whatever design automation problem we are going to solve, we need to have a sequencing graph, and then we talk about the design flow. As this is this has the similar analogy with VLSI. Like in our VLSI, we write the program for adder in Verilog language and then synthesize it in our Xilinx software. So like this, you will have some program to write. So that is your sequencing graph model. You can use the gate level notation also for that in VLSI chip. So you can also use the behavioral notation also. So that's why similarly you have to basically write down your input file, the program, and you have a software. You have to do the design automation with the input, this input file. Okay. And as an output, what you want, so it is mentioned here that you want two things basically. So one is this the layout of the chip. I have shown here a very simple one, that is 6x7 array, but this layout may not be always regular shape. It may be a polygonal shape, orthogonal polygonal shape. Okay, so if you are dealing with only square shape electrodes, then you can have an orthogonal polygonal shape of the layout. You can have the hole inside the layout also. You can have a very irregular and different layout also. But you have to define that what will be your program, your EDA tool, what is that called, the electronic design automation tool, will give that layout. Okay, so given the graph, you have to design, you have to get that layout output as the output. And another thing you also have to have is that actuation sequence. So if you follow these electrodes, so you have 6x7 array, that is 42 electrodes. So total 42 electrodes you will have. If you imagine that everything that very simple, like in this case, to understand what is the problem. So here basically you are considering only digital microfluidic biochip, but in case of continuous flow microfluidics, you will have the valves, and those valves are also individually controllable. If you are if you are thinking like that, here all these electrodes are individually controllable, and in that case 42 electrodes, you will have 42 bits for each timestamp. So you can see that at every timestamp, what are the values of these 42 bits. So that will give us that this is the binary sequence, which is also called as actuation sequence, basically, to be applied to those electrodes at that timestamp. And the next timestamp, you have to apply the different binary string. So like this, it can be viewed as simple as, like we talk about this in system software, that we have the machine language, and this is basically binary bit string, and this is being loaded into the RAM, and then it is executed one by one by the through the clock cycles that is applied by the processor. So similarly, you can imagine that you have a microcontroller which is having this memory with it, and it is loaded with this actuation sequence, and the microcontroller has the control pins, which is connected to the microfluidic chip, and these controls are being sent. Control sequences or the actuation sequences are being sent there, clock cycle wise. So I'm taking the similar analogy with the VLSI chip design, which is design flow we studied in our VLSI course or computer design course, electronic design automation course, whatever. So basically, we are taking the circuit specification as in C++ or maybe System C or Verilog language, and then what are the different phases we follow? Basically, high-level synthesis first, and then physical design. Broadly, if you say, in between we have the logic optimization and logic synthesis also. So in the high-level synthesis, we have this scheduling problem. So we have the directed acyclic graph. We have to schedule that, and then do the module selection. So you have module library also with you, and then you know that which module is taking more time, which module is taking more area. So you know all the details. That's why you're selecting the modules and binding the modules to these operations in the scheduling graph. So that's called basically the high-level synthesis, and then the logic optimization comes, and then system partitioning. So partitioning problem is solved in the physical design automation. So here we'll come to the geometry of the chip. So system partitioning talks about that where will be the different set of operations to be done. So you can, whether you can cluster them and you group them in a particular region of the chip, not just randomly placing in different locations. So if you can make them clustered because of depending on different other overheads that will be there, so you can partition the system means on the chip geometrically at different locations, different portion of the layout, and then floor planning. So that means you can orient the, maybe you can say that you can change the orientation of the block, microfluidic chip block, or these partitions, and also you can place. So you can place in different way in the within the same region of the chip layout. And this placement, after doing this placement, you have to deal with the pin assignment. So you have to control the electrodes. It is better to control by individual voltage, but it is very difficult also. It is not very easy, because when the chip size increases, then it is very difficult to wear out the wears from the center electrode, because each and every electrode is very small, and you have to have some dimension of the wear. So wear is not so so small that you can make the width of the wear is so small that you can make so many wears out from the chip. So that's a basically a different problem, physical problem. So that is dealt in the physical design automation. And then people talk about this pin constraint based biochip. So that was discussed in the previous lecture also, Professor Chris. So there are many algorithm people have proposed that how a particular biochip can be controlled with less number of electrodes, less number of control pins to control the huge number of electrodes. Okay, so that's an optimization problem also. So broadcasting based algorithm that was discussed yesterday. And routing algorithm is here, basically routing problem. We are talking about, okay, I may be mixing both, but maybe these steps are similar. So in VLSI chip, the routing means the channel routing and the detail routing. So these routing means the wear routing, but in case of biochip, it's basically droplet routing. We have seen the things, what are the problems here. So finally, we are going to have this layout of the chip or the mask in the VLSI chip, which is written in terms of GDS2 file. Similarly, in biochemical process automation, like you can say the microfluidic biochip automation, design automation, the design flow follows the similar kind of steps. Like it takes the bioassay description. Till now, I was talking about the problem that how to standardize the language. That is one problem. And even if we think that we have the DAG, somehow we got the DAG, then we can proceed with the similar flow that we do first the high-level synthesis, and it involves that operation scheduling, resource selection, means the module selection from the module library, and binding them properly. Okay, so based on the number of module available of same type or different type, you can choose the different possibilities. So there's an optimization problem also. There are many different problems people have solved by solving the optimization problems for scheduling, binding, and all these things. And then the module placement is a geometrical problem. So you have the module, different size, different their attributes, like time taken by that module to mix particular set of fluids, or maybe you can say that what will be the size, means area taken by that module based on the dimension of the module. So this module placement takes care of that which module to be placed at which location geometrically on the layout. So that basically optimization problem. So you have many possibilities to do, and you have to find out the optimal one based on the different optimization criteria. So that's basically dealt in the physical design automation process. And droplet routing, as I told earlier, the droplet movement from module to module, or input to module, or module to output, like this. And pin assignment, I was talking just when I was discussing this, that the different pin constraint biochip has come, and they are solving different optimization problem, how to minimize the number of pins to control the chip. So final outcome is similar, just one thing is extra, that is actuation sequence. So layout is always required here because the geometrical problem, it is geometrical layout of the chip. And basically you have this layout of the chip along with the actuation sequence of the electrodes. In case of digital microfluidic biochip, if you are talking about continuous flow microfluidic biochip, then you are basically dealing with valves and the pumps. So pumps are basically made of 3 valves or more valves, and sometimes basically, so in general, you can think that all these are basically controlled by the valves. So in case of continuous flow microfluidics, you will just tell that actuation sequence of the valves, micro valves, and in case of digital microfluidics, you are telling that actuation sequence for the electrodes. So the process is similar. So similar kinds of steps or the optimization problems are being solved. So there are different optimization criteria people talked about, and in different papers you can find in the literature. This was already discussed by Professor Chris. So maybe in short, I can say that you are given the sequencing graph, you are given the module library, you are given the design specification by the user. Okay, so here these 3 are given, and the synthesis process of the design flow is giving the output like the resource binding, like which operation to be done on which module. So that is to be decided, and the schedule, that is basically the timing. Okay, so which operation will start when, and which that operation will end at which time stamp. So this timestamp is decided first, that how this clock cycle is decided based on that, and then this different operations are scheduled based on this schedule, or you can say that placement and this schedule both are simultaneously being solved, and then you are getting some kind of result where you may see that there is an overlap of 2 modules in the placement result, but these 2 modules are not being executed at the same time. So once one is completed, then other one is starting. So that's why it is possible to have overlap on that. So as that very nice example was giving Professor Chris by railway crossing. So you have the crossing possible, but this is not the same time they are coming there. Okay, so at different time they are being used. Okay, so the fluidic constraints, this is important important constraints. These are important constraints related to the droplet routing. So because we do not want to make any unintended mixing during the movement of the droplet. So the droplets are sitting in the mixer, then they want to mix. So that's why we can steer that. We can make the control sequence within the mixer in such a way that they can merge and get mixed and give us the homogeneous, or you can say the uniform concentration merge droplet or the mixed fluid. But in case of unintended mixing, when we are moving the droplet from this location to that location, then we are not intended to mix that. So how to resolve that problem? So you have to solve the optimization problem. So there is a droplet routing problem, which is an you can think as an optimization or automation problem, like how to send the droplet from following which locations. So the simple idea is this, that there should be a gap of 2 electrodes. And similarly, the other rules can be defined also for different phenomena. So I'm just telling that the simple gap, simple idea is there, keep 2 electrode gap. So then they will never get mixed. But in case of this kind of scenario, maybe they get mixed. So that's why in that case, another constraint is required. So that's basically the it was discussed in this paper also. Based on this fluidic constraints, the different droplet routing algorithm has been proposed by different researchers, not only this paper, but a lot of papers have been published on this droplet routing problem, considering different application, considering different you can say the type of chip, like pin constraint chip, how this droplet routing can be done. So this based on the different applications, different droplet routing strategies you can find in the literature. And they are using, like, sometimes they're using A star based algorithm in AI. We know that it's being used in the planning. And also sometimes this geometry based problem they solved. Okay, so this is a simple idea. Another idea, that is when the mixer or some module is being used, then any droplet which are being routed from another location to, say, some another location, not intended to mix with this mixer module, then we should have a guard ring. Like we make those electrodes forbidden, like those are obstacle. So we do not want to move our droplet through those electrodes. So that means there will be no unintended mixing happening with the droplet which is being routed. Okay, so that's basically another simple idea that people have used in literature, that they have used the guard ring for each and every module. Okay. The contamination issue was also discussed, but yes, so as we have seen that if we change the routing path, then we can reduce the contamination number of contamination cells. Okay, so you can see these solutions. So there are three different source droplet. They're moving to some three different target locations. If you change the routing path, then you can reduce the number of overlapping electrodes. So finally, this solution is telling that this is basically having less, only two overlapping electrodes. So people, when talked about this contamination, ever droplet routing, they also talked about washing of these electrodes, because these are getting contaminated. Contaminated means some small portion of the fluid which was being sent through that path, through that electrode, maybe dropped as a residue, a very small amount, and that contaminated electrode need to be washed. So people are talking about that, how this droplet, which may be water or some other buffer liquid or washing fluid, that can be used to wash this droplet. Although this washing is not a very widely used, but maybe some of the routing algorithm and contamination routing, we will talk about washing also. Okay, so till now we have seen different design automation problems people have addressed, and there is a group in University of California, Riverside, USA, by Professor Philip Brisk, and they have developed a simulator. So simulator is just like they are taking that protocol in terms of DAG, direct acyclic graph, as a text file, simple, and then they read and then solve these optimization problems for module placement, module routing, droplet routing, before that dispensing and all these things, and then detection, dilution, all this storage operation. You can see here some storage operation also there, mixing operation there, dilution operation there. So they are performing these operations on the chip at different timestamp. So this TS is timestamp. So they're changing the timestamp, and you can see the simulation of your algorithm. Okay, so you can design different algorithms and use the simulator to test whether this algorithm is giving better results compared to the previous one, or you are doing better or not. So that's basically a simulator for digital microfluidic biochip we can use. It is known as DMF SSS, I think static simulator, something like that. Yeah. So one hardware kit, basically very popular. I like their research basically. So basically they did some small kind of their development. Basically they have done some development work, and they're selling this kind of kit. And these kind of kits are basically having a microcontroller inherently, means at the bottom of the chip. So there is a whole kit, and this microcontroller is based on Arduino kit, and this is the older version. In 2015 this version was there, and and this is 2020 version, and it is basically version 4. So this is the latest one. So you can see both the version. So the 5 years ago what they were doing, so they have the simple this electrode array of electrodes. They were putting the drop fluid on chip with the pipette, and then doing this movement. So they are selling this kind of chip. They are also selling nowadays for experimental. You can say that for the university research purpose. So if you are interested in developing this kind of algorithms and to test them whether they are performing properly for a particular protocol, you can also buy this and then do this on the chip to test whether the algorithm is properly working. So this is for digital microfluidic biochip. You can buy this kind of chip. Maybe I like their development. So there are a lot of advancement they're doing. So this is one thing to share with you, that from Godila from Switzerland. And now I'll talk about some advanced microfluidic biochips. So initially we are talking about maybe 5 years ago, we are talking about only digital microfluidic biochip and continuous flow microfluidic biochip, 7 years ago. And after that, we got these new advancements of the different digital or flow-based microfluidic biochips. So I will go one by one. Maybe already some of them are touched already. So I will tell how they are basically. So MEDA is one that was published in. Okay, so first one thing to mention here that the very good journal, this Lab on a Chip, is very good journal and very high impact journal in which this kind of new technology which are being developed in research lab or mostly research lab, they are being published there. So they are basically showing, demonstrating that this is the new technology, how they are working, and how basically the programmability is possible or not. So Lab on a Chip is talking about that and which application they are targeting like this. So if we look into this paper by Hedwin, it talks about this MEDA. So MEDA is microelectrode dot array based DMF. It is the digital microfluidic biochip, but they made very small size microelectrodes to make a big electrode. So basically, its granularity has been increased compared to the digital microfluidic biochip. The another one is this PMD, this programmable microfluidic biochip, which is also this is not digital, but it is flow-based, but it takes the similar architecture like grid-like architecture as digital microfluidic biochip is having. In continuous flow microfluidics, we are generally seeing this circular mixer and this irregular shape of these channels and all these things. But they, first time I saw in literature that they have talked about a grid-like, a regular size of the continuous flow-based microfluidic biochip. So this was done in EPFL, I guess EPFL, yes, Professor Sebastian Merkel, and it was published in Lab on a Chip journal again in 2011. Then another advancement that was commercialized basically from Korean University, Sogan University, Professor Kanho Sen and his group, they come up with a startup company also, which is basically taking this, you can say that electrodes is being fabricated on paper. It can be fabricated on the newspaper also, and they are using carbon nanotube material to fabricate the electrodes, and they are controlling the electrodes by external voltages. And they publish some papers also in Advanced Materials Journal in chemical engineering. So they're from chemical engineering department, and they have commercialized some of the chips also in their country and outside also. And random microfluidic biochip, as it was discussed yesterday, I think Professor Ho was discussing that machine learning based techniques for this kind of chip design. So this was also published in Lab on a Chip journal by Wang and et al. And Professor Philip Biss was there from University of California, Riverside. And they published this kind of architecture where they basically also talk about regular size electrode, but it's a random regular size. Basically, you have the regular grade of electrodes, but you are switching off some of the, not electrodes, it's channels basically. And you are switching off some of the channel arms and making it irregular shape. So starting with regular array of channels, you are switching off some of the, you are deleting some of the electrode arms and getting some random continuous flow-based microfluidic biochip. So what is the use of this microfluidic biochip? They have shown for dilution example in their paper. So maybe if we go with this video, some of the properties can be explained, like in MEDA, microelectrode dot array DMF biochip. So in that case, if you see that there are microelectrodes considering one electrode, so you can have more granularity of the fluid control. So this has given a lot of new challenges to the DMF researcher. The people who have worked on this DMF digital microfluidic biochip, they got new problems, new design automation problem, new optimization problems when they dealt with MEDA biochip. And basically, you can see in this video that the fluids can take any shape, not only just circular shape or the shape of the electrode. You can change the shape of the fluid, and you can also perform different kinds of mixing and splitting with different volume. So you can control the volume further, right? In case of digital microfluidics, you can have only 1:1 mixing model. So I'll talk about that little bit in the sample preparation. So here you can see that you can make different varying volume droplet from an on-chip reservoir. So you can imagine that there is an on-chip reservoir having huge volume, and you can allocate. It's called allocating of different volume fluids, droplets. Okay. And you can also see that it is also possible at diagonal movement. So earlier, when people used to talk about this droplet routing in digital microfluidic biochip, they are talking about this Manhattan type movement, like either horizontal movement or means a vertical movement. But here you can see that this is also possible to make a diagonal movement, which can reduce the droplet routing time. Okay, so that's why people talked about many different new algorithms based on this microfluidic biochip for the similar kind of problems that they address in digital microfluidic biochip, the traditional digital microfluidic biochip. Then I was talking about this PMD microfluidic biochip. It is also named as, you can see in the literature, people have also called it as FPVA. FPVA is basically fully programmable valve array. So you can see there is an array of valves. So these are the area, these are the valves, and this is the cell within which the fluid can be kept. Now you can switch on and off these valves in a circular pattern, and you can make a mixer within this, as it is being done here in this video. So you can program these valves in such a way that you can make a mixer within this regular shape continuous flow microfluidic biochip. Okay, so this is a very interesting advancement. You can say that was done in Switzerland, this EPFL University. The third one that I was talking about this is the paper-based microfluidic biochip. It is digital microfluidic biochip, and they are from chemical engineering. This is Professor Kanho Sen, and they did this kind of chip. So they have fabricated, they can print the chip on paper, and they can using this carbon nanotube material, and they can control the electrodes. So they have made some commercialization of the product. So of this kind of means paper-based microfluidic biochip. So when this kind of design, or this kind of new technology has come, so many people, many researcher, design automation people, or from the computer science people, you can say that we got some new problem to work on. So some design automation computational problem was defined, and people have published some papers on this. So okay, so this is one advancement for third one. Then the fourth one is this random microfluidic biochip. It's already we discussed. I think Professor Ho discussed. So this is having this array of channels. You can say the regular kind of channel started with regular channel, and then removing some of the channels to get a regular irregular shape. And you can say that's why they are calling this as a random microfluidic biochip. And they tested this for their research in this journal. They tested this for the dilution problem. So they are generating different kinds of dilution concentrations after sending the sample through this inlet and the buffer through this inlet, and having 3 different outlets in their experimental work that they did. And they talked about many different computational problems here, but like there is a huge number of possible design. So which design to be considered, and how to know that which concentration will require which kind of design of the layout. So that's the computational problem they introduced in their paper, and then people worked on this problem, and it has basically a huge database that is to be searched in to find out the optimal solution. So there is a scope of applying machine learning. That's why people worked on this kind of chip design, applying some machine learning based techniques, artificial neural network based techniques to optimize the design. I will talk about basics of some automated sample preparation using biochip. So why the problem is coming. So in my one way, I want to say this has been discussed by other researchers, or other speakers. So what I want to say here new is that, so when you are doing this sample preparation in the laboratory, you are given some supply of fluids. So you are dealing with dilution problems. So you're only having 2 fluids. So this yellow and blue, and you know that the total volume is 22 ml and the test tube volume is 100 ml. So you know that at a time you can take 13 ml of one fluid and 9 ml of the buffer, and then mix it by the steering stick. So this is possible. But when the total the ratio that is asked, that is being asked by the user, that you have to prepare this ratio, and which is of maybe 50 ml only, and you are not given anything else, only this test tube, no container. So you have to think little bit how maybe you are given only one extra test tube, like your tower of Hanoi problem. So you can think like that. So you have given one test tube, then how you can mix them, these two, in which step you will follow to discretize the different steps, and you will finally achieve this ratio 71:49, which is actually total volume. If you directly mix, then it is 120 unit, 120 ml volume. If you take the test tube of this 100 ml, then it is not possible directly in one step. So that's why you have to decide, you have to solve this step. So you have to find out what are the steps. This is the optimization problem. This is the basically automation problem. So that was dilution, and in case of mixing, the similar problem you can think of, that you are given supply of 5 different fluids of different colors shown in this image, and you have to optimize the number of steps to achieve this particular ratio of them. So you want to mix them at a particular ratio. At the end point, you want this of this as an end result, you want to have this mixture of 80 ml volume. So then how you will do this? Well, means you are given maybe only one test tube, or maybe 2 test tube. Depending on that, you can think of optimization problems. Okay, so so similarly here in digital microfluidic biochip, when you are going to automate the sample preparation on this chip, you are supplied with sample, and sample is generally thought of as the 100% concentration because it does not contain any buffer in it. So it is pure sample taken from my body, that physiological fluid, maybe saliva, maybe blood sample, and all this. So this is 100% concentration, and I have to dilute this because of the bioprotocol that I want to implement, or I want to perform to test my say glucose measurement, or like other things, a lipid profile or so. So you are given the buffer. So buffer does not have sample in it, so that's why it is considered as the 100% concentration buffer. But in terms of the calculation here, we'll tell this is 0% concentration because it does not contain any sample in it. So that's why we tell that 100% sample and buffer is 0%. And the target concentration may be given in terms of a fraction, in terms of percentage, in terms of ratio, any way it can be given. That is CT, that you can imagine that it is given. You have to generate basically this concentration of the sample. And you are also given some concentration error tolerance. So if somebody has told you that 33.3% concentration you want to you have to prepare, then maybe it is told that you can tolerate up to 0.05% means like error. So that's one thing. And the volumetric constant, that is mixing volume constant that I told in the previous example, the 100 ml. So that is the volume constant that is given. So based on different architecture, this volume constant matters basically. In case of DMF, it is 1:1, so total volume is 2 unit volume. In case of maybe MEDA, it is more. So you can increase the volume shares more than 2 unit volume. And the objective is to optimize the number of steps, the mixing steps, and also you want to reduce the uses of the fluid and also reduce the wastage of the fluid, because if you are creating more and more wastage in the whole process, in the intermediate steps, you have to move them out through out from the chip to the waste reservoir, so that you can get more space to deal with for the routing of the droplets for the next few operations. So these are the objective basically to find out what is the output. So output is basically that sequencing graph, that DAG. So DAG we have seen earlier. So we have to find out this kind of DAG, that is sequence of operations. In terms of a graph or in terms of a tree, you can represent that. And you have to generate that given this input and given these objectives. So this is basically optimization problem. Sometimes many times people have talked about this in terms of optimization problem. So I'm not going into detail of much of this. Maybe I want to come, so this is already discussed. Dr. Topolina has discussed this, that if you take this concentration, you convert this into binary scheme, binary string, then you read from right to left, then you get the tree or the relative acyclic graph for generating the target concentration. And this was done very old in Natural Computing Journal 2008 from MIT group. And then we talked about something, some algorithm in 2010 based on this binary search-based techniques, and which gives better performance in terms of low consumption of the fluid, and also the means reducing the wastage, basically reducing the wastage of the fluid. If you see this histogram, so this is red one, that was previous one. So this was talking about many number of concentrations are having this more waste, but we got less waste. So we published in TCAD journal in that for that work. And then mixing problem people have addressed in 2008 in this journal is similar to that binary bit scanning technique. And it's having the 7 different, in this example, we have taken 7 different reagents, and they are developing this tree bottom-up fashion. So they are getting these different fluid identifiers at different levels. If there is 1, and if there is 0, then that level is not coming in that particular level of the tree. So if you develop this tree in bottom-up fashion, then finally after scanning these 7 bits, you will get the tree. And this is called, the algorithm was named as a min mix in that paper, so we called it as a min mix tree. And we proposed another algorithm that is a partitioning based, number partitioning based technique, and dynamic programming based approach we applied. And we have shown in this paper in 2015 two days transaction in ACM transaction for design automation. So we have seen that different dilution subtrees we can, if we have more longer dilution subtree. What is dilution subtree? Because this subtree is having only 2 fluid. So if only 2 fluids are being taken, so you can compartmentalize this mixing operation, and you can have less overlapping of these electrodes. Means you can say that overlapping of different fluids in a single electrode, so that will cause less contamination, or the routing will be easier. So like this, we have explored this algorithm, and we found that this is performing better. And this was a top-down approach. The earlier one was bottom-up approach, and this is top-down approach. And we found this tree in this way. That algorithm was discussed in the paper, in today's paper, and the performance was shown based on their layout awareness, like how this mixing tree is aware about how this tree will be implemented on the chip. Because the previous one was not so concerned that how that tree will be implemented on the chip. But we talked in this paper about how this tree will be implemented on the chip. In terms of that, this algorithm was performing better. And then in 2013, we in a conference we published another paper which is talking about this sharing of this waste droplet. So if we share this waste droplet, there are basically 1:1 mixing you are performing, and you are getting 2 unit droplet after splitting, and one is used here, so another one was going to waste. But if you are not, in the previous algorithm also it was happening, so in the mixing algorithms, but if you are not going to waste this, you are going to use that, then you have to get a similar kind of subtree structure in the same tree, so that you can reuse this. So we proposed another method that that was talking about this, that how we can get a similar subtree, common subtree within the same tree, so that you can use the reuse the previous mixture or the intermediate mixture. Okay, maybe I'm running out of time. So quickly going to some of the algorithms people have talked about, many papers. There are a lot of papers people have published in this domain of automation of the sample preparation. The people talk about in terms of Fine's taxonomy kind of nature, like we have seen in the architecture, computer architecture, Fine's taxonomy. It talks about single instruction, single data, multiple instruction, multiple data, single instruction, multiple data. So similarly, like that, if you have a one target concentration to achieve, then single T, and demanding only one droplet, then single T, single D. So STSD. And then similarly, you can think of that people talked about in these papers, in all these papers, you will find this different category of work. So they are having multiple target, multiple demand, multiple target, single demand like this. So recent results in our recent papers, like one way, I think I should skip this, it will take a longer time. So maybe I will talk about this, this one in last year. So we talked about this fluid transportation in FPV. FPV means this PMD, same thing. So we have basically different type of operations, as we can see that fluid loading is important here. So if we look into the loading problem, okay, I will come to that loading problem later. So maybe if we talk about that mapping of this mixing tree on this kind of chip. So this is a PMD chip, which is a 2D grid-like architecture, having this array of valves, and it's fully programmable. And you can model this, you can map this tree into this graph so that this mixing operation can be done. As we have seen in the video of PMD, this mixing can be configured by sequentially controlling the valves within this circular mixer. So we are talking about 2:2 mixer here, because we have 1, 2, 3, 4 cells. So 2:2 mixers, how these can be executed on the chip. So based on that, we propose some technique that first modify the mixing tree. That is one algorithm, that heuristic distribution algorithm. And then this modified mixing tree is taken for mapping. So this is a transport-free mapping because we do not want to move the droplet, move the fluid from one mixer, which is continuous flow, fully programmable mixer, from one mixer to another mixer. So that to avoid that, we propose another scheduling technique, and then we got the output in that way. So in this work, we have talked about two things. First one is this one. So it is the previous mixing algorithm that we got, means the mixing tree, as we have seen in the min mix algorithm. So if you directly map this one, so then different valves will be executed means many times. So it is also means, as we have discussed in the previous talk, Professor Krish and Professor Ho, that the valves, if it is multiple time it is being used very frequently, then there will be some degradation also of the valve control. So they may degrade it, or there will be some problem with the valves, so they may not be working better way. So we need to distribute this valve actuation over the chip. So when we distribute the tree in different way, this is another means you can say that it is using the similar same fluidic consumption. It is doing the similar, almost nearly similar amount of fluid being used, but the valve actuation has been distributed. So we talked about this method that in this paper, this distribution algorithm, and then no transport mixing. So that is mapping basically. So given this mixing tree, you can see the different color notations here. So M5 has been modeled here, and M2 is mapped here. So both can be executed at the same time. So these different trees are loaded, and then they are mixed. And then M4 is being loaded and mixed. And you see that you are using this one droplet from, M5. So M5, from M5, you are using one droplet. So you are, after using this one, you can get this M4 to be done. And then you are using this, you are going to do this M3 operation, and then finally M1 operation. So without any movement of the fluid, how we should place these fluids on different cells in the PMD chip, that is a challenge basically. How to place these different, what will be the different optimal location of the fluid in different cells. So after, so this was on PMD kind of this advanced microfluidic biochip. What we did, that is mixing work. And then another one was on dilution, and it is for MEDA kind of biochip, so MEDA. So in MEDA biochip, we have different mixing models. Not only 1:1 mixing model is allowed there. So different mixing models are possible. And we talked about a different design flow, where we basically talked about first find out the optimal number of optimal order of you can say the numbers for generating this tree. So after maybe this will take maybe you can refer to this paper, because here actually we are talking about different you can see here FLOSSPA is one existing paper, and this is WSPM, another paper. So they were taking this 5 size mixer in all the mixing steps. Here also 5 size mixer in all the mixing steps. So here we talked about this factorization based dilution algorithm, where different factors. So these given the total denominator value, that is here, that 240, this is the target concentration. So 240 can be factorized in different way. So prime number factorization, or you can say that prime and conjugate number factorization, composite number factorization. So we dealt with that. So different type of factorization. So 3, 4, 4, 5. So different factorization we used, and then come up with better solution for meta chip. So that's basically our recent work in 2020 and 21. And we worked on this loading problem of PMD biochip. So loading problem is as simple as this, that when the fluids are loaded in different cells, if you see the video of this paper, you will find that the cells are loaded by sending the flow from top right corner inlet and bottom left corner of as the outlet, and different cells are being loaded. So here is the problem, that when we are going to map the mixing mixing tree on this chip, then you have to first determine that which cell will have which fluid to be loaded. So loading is the important problem in this kind of chip. So we dealt this problem as in 2 parts. So one is that how to decide that which fluid, different colors have been used for different fluid. In a particular bioassay, we have considered here 3 different fluids are there, and they are being loaded into different cells. So first you have to decide that which cells will be loaded with which fluid, and then how these loading cycles, means flow, means how minimum number of flows you can have to load all these fluids. Okay, so it is not possible in one flow you can load all the fluids of the same type also sometimes, because there is there are obstacles with other fluids. So you have to deal with multiple flows to load the cells with the different fluids. So we dealt this problem in different phases. One is this LAFCA, that is basically layout error fluid loading, loading error, basically fluid assignment, fluid to cell assignment problem. And we dealt with this problem in this way, that different cells are targeted for different fluids, and then which one is the optimal solution, we found that. And then for the particular problem, when this is decided, then fluid loading cycles are decided. So how many sequence you need to follow, flow sequence to follow to load the fluids. So I'm not going into detail. So in conclusion, maybe these are the basically recent work that we did. So basically these are the work papers you can find in the literature also. So in conclusion, maybe we are looking towards a scenario when there will be a microfluidic biochip in our hand, and we have this smartphone, and we can download the Bioprotocol from the Google Play Store kind of thing, or maybe even in the Google Play Store, you can download that glucose test app. You download, and you get the actuation sequence for the particular architecture you have in your hand. So for this architecture, you have that, and then you send the signal through the Bluetooth connection, as they are doing here in Washington University in lab. So basically, they have this Bluetooth connection to control the biochip, and they're downloading the protocol. They're taking different protocol. So people are looking towards this kind of smart, you can say, the healthcare device. And like we are using smartphone, we'll be using sometimes the smartphone, and this will download different applications, and this protocol will be tested. Basically bioprotocols will be run, and biochemical test or pathological lab test will be at our own end. So we can do that by authorized protocol to be used. So there are a lot of challenges here. People are so we want to see the better for the betterment of our society. We target this. Our target is to develop low cost, portable, automated biochemical laboratory on chip, and we want to conduct this research. Basically we need different disciplines to come together, so it's a multidisciplinary research. And the main recent thrust is towards this rapid, what I feel is this important is this fabrication and fabrication process that basically to be because the industry, when this will become commercialized, more industries will be interested, then the fabrication will be a very streamlined process. And then this rapid prototyping of different application-specific microfluidics biochip. That's the main research thrust nowadays. And the cyber-physical integration for the automation of the chip and the users of the chip is also important. So people are doing this research in this direction. So thank you for your attention. I have crossed 7 minutes time. Okay. Yeah.
Thank you, Sudipda, for sharing with us such an informative talk. You are really take us in the world of microfluidics in such a way that the time is not the matter. Thank you. And the way you explain the design flow comparison between VLSI chips and the biochips, it will definitely trigger research idea among our audience. So, am I audible?
Yes.
Okay.
Yeah.
So I request our participants, please come up with your questions.
I have a question, sir. Actually,
Yes.
actually, the question, maybe I am actually actually the new actually in this field. So from the last 2 days talk and from today's talk, actually, what I understood from that, I am asking you the question that that unified synthesis that you have just give the overview. So there you have said that there will be a sequence graph, my digital microfluidic module library, and the design specification and the resource binding. So yes, sir, that slide actually. So here my question is that depending upon all these four stages, like the sequence graph, digital like get or module library, and design specification and resource binding, all these are given by the user, or these are also the part of the synthesis process?
Oh, okay. No. User is giving this assay, basically which application you want to implement, and the module library, as a designer, we are designing as a designer and taking that as an input, because I will give the analogy also, and the design specification is given by partially by user and partially by the designer. So how it is, because if you see this maximum bioassay completion time, it is basically dependent on the application. So if you are taking the sample and you are waiting for longer time to get the result, maybe the sample will degrade. So that is basically application specific, and the user knows that. So user is giving this time constraint, but as a designer, I am deciding, okay, also this area. So area, maybe the application designer or application user knows that I don't want to make the chip larger, very larger. I want to keep it in my wristwatch size device. Okay, so that's why this array of electrodes is decided 20 by 20 is the maximum size. Now this is also, you can say that this is taken from the user, but this maximum number of detectors, this is by the designer. They can decide that how many detectors I want to keep, because once one detector is being added, the cost of the device will increase. So whether your application can be implemented with only one detector, or maybe it requiring two or more, that the designer will decide. Okay, so based on the application. And similarly, the number of reservoirs, this is actually given by the user only, because user knows that what are the different reservoirs you need. So input device, how many input reservoirs you have, input port, and output port, you are deciding as a designer, because you know that how many fluid droplets you will generate as waste droplet, and you will send those out from the chip to the waste reservoir. So if you as a designer thinking that, okay, I need to wash out more and more fluids because I will not get the space available, in that case you will have more number of waste reservoir maybe, or maybe some another arrangement. So depending on these design specifications, so that's why it is written in general that this specification may be sometimes depending on the application and the user's perspective, and also the designer's perspective. But this design library, which is basically the designer itself is deciding. So designer knows that on the 2D microfluidic, two-dimensional array, what will be the different mixers can be reconfigured, and what is the size, means number of cells based on their dimension, and the time they tested the different fluids they have taken. So they have taken different fluids and mix, and then see that after how much time this homogeneous mixing has been done. So based on that time, they have recorded all this, the designer. They are also taking the help of how they will do. They will take the help of the experimental laboratory, like wet lab. They will take different fluids and then mix and then decide the time that after how many seconds this is a homogeneous mixing or uniform mixing. The concentration is uniformly done. So that's based on that, this time is decided. So this module library is actually taken as the input. So the design synthesis, unified synthesis, taking all these as the input and then deciding the output is binding. And this requires algorithm because there are many possibilities. Okay, and the scheduling also. So time when to start what and this placement also geometrically doing this.
Okay. Thank you so much, sir. Thank you so much.
Yeah, thank you.
Okay. We have 2 more questions. Please.
Okay.
Yes, a very nice talk, sir. I just want to ask regarding, you have talked regarding the automation and optimization problem. There's you have basically talked about the DAG, the direct acyclic graph. So there, like if we want to, there may be so many solutions for the DAG, and if you want to study the cause, what will be the cause of the solution, and also I may come up with multiple solutions. So is there any simulation tool or something that I can study, like how do I know I get the optimal DAG that meets my constraint?
Oh, okay. So you're talking about this DAG, directed acyclic graph, this one.
Yes, sir. Regarding the automation and the optimization, there for the sample mixing, you have given some.
Okay.
Yeah.
Sample mixing. So there are basically 2 things. One is this DAG. This DAG is basically talking about the bioprotocol. And this is basically talking about the actual bioprotocol that will be implemented, and that is being done in the biochemical laboratory. So biochemist tells us that these are the different input fluids to be taken, and they will be mixed in this sequence, and this is the output I want. From these, we need to draw, or maybe they can draw the sequencing graph or the directed acyclic graph. But the other problem that I discussed in this talk is that the sample preparation, where you are told that concentration, I want this one, and this is the architecture I want to use, whether this is DMF or it is MEDA or it is PMD or the continuous for microfluidic biochip. So depending on the architecture, you know that what are the different mixing model allowed. And that time you are basically developing algorithms to generate this mixing tree, basically determine the mixing tree, or that is also a directed acyclic graph. You can imagine like that. And in that case, the optimization criteria are different, like how fast you can do the sample preparation, like mixing or dilution. That is one criteria. You have to do it fast. And the second criteria is you have to reduce the sample or buffer or reagent uses. And the third criteria is the reduce the wastage. So you should not waste the reduce. So depending on the application, your optimization criteria may vary. So actually, we worked on this problem little bit in general way in a work in PMD. You can see that paper in which we talked about this different optimization criteria, and those optimization criteria are coming from I think this paper. You can see this paper. Here we talk about different optimization criteria. This one. This today's DPMD. This is for PMD kind of chip. So based on the different optimization criteria, you decide that whether this directed acyclic graph or the mixing tree or dilution tree is optimal or not. Actually, it depends on the user's perspective, and also the optimization criteria. Those are fixed for that particular problem or application specific chip.
Okay, regarding this mixing, as of now, what I have understood, so there is no such tool that can measure, so like we may have different mixing tree or different multi-objective optimization. Maybe we want to reduce the number of reagents or to have a better solution. So as of now, there is no such tool, right, sir?
Yeah, actually, the people have worked on these problems in different way and worldwide by different groups, and they targeted different architectures. There is till now there is no such means industrial or maybe that open source tool, you can say, that where you can just choose the architecture and choose the mixing model and then run that algorithm and test it with the competitors. competitive algorithm, means representative other algorithms people have done. So that is a thing that maybe development work people maybe they can target to do this. It is important. It's, I one time I started with some of my students, but I could not succeed. But yes, that is a requirement. Yes, one particular tool, GUI based tool, is better to have, in which means different architectures can be taken as the input or considered, and then you can choose that which architecture you want to focus, like PMD or MEDA or DMF. So, and then you can choose the mixing models also, and then you can decide, you can implement your algorithm there and test whether it is performing the better performance in terms of optimization criteria with the competitors.
Yeah, yes, sir. Thank you.
Yes, right. Thank you.
Thank you very much, sir, for such an elaborated and talk. Myself, Piyali Datta. Actually, my question is that in PMD, you told that there is one upper limit above which, if a valve is being activated and deactivated continuously, it may be degraded. So, is there any particular value that above which the valve can be degraded?
Yeah, actually, this PMD was developed by Fidalgo's group, Fidalgo and Professor Sebastian in Switzerland, EPFL, and they have published this paper. Along with that, they have also means given the supplementary material where you can find the details of the materials they used. So the question that you are asking, that the number of valve actuations, that basically depends on many parameters, like what is the material used here, and how is the you can say the switching activity of the valves. So depending on these, all these, based on the clock cycle which is being used here to control the valves, so many parameters are there. Exactly, no number has been mentioned in that particular paper, but it is from the common understanding that when you are dealing with some valve actuation, and also maybe other papers, not in this paper, who talked about this valve actuation based reliability aware chip design. I think Professor Ho was discussing, he published
Yes, sir.
one paper. So reliability aware chip design. So in that case, the valve actuation, so number of times the valve is being actuated, based on that the degradation of the valves happens. So you can for the sake of development of algorithm, you can decide a particular number as a fixed value, but it comes from the design specification or even technology of the particular fabrication technology that is being used. So that will be
That's
given. Yeah.
Yes, sir. That's why when the jobs were distributed among the valves, we have to take a particular value, say K, and afterwards we can have that value from the designer. Yeah, is it like that?
No, actually, before you already decide the mapping of the fluidic operations on the chip,
Yes.
you have to take care of this number of sequence, because the problem is to make reliable chip, a robust chip, so that it cannot it do not basically over exceed the number of this threshold value of
Yes,
the
sir.
actuation. So before mapping it, you have to consider this.
Consider that
Yeah.
value. Yes, sir. Sir, another question is that in case of say MEDA, if some of the microelectrodes are degraded, say those are faulty, is there any problem in droplet movement in that case? Because a droplet is held on multiple microelectrodes. If one or two of them are faulty, will there be any problem?
Yes, there will be problem, and that I think Professor Chris has already discussed this topic. I saw in the yesterday's
Yes.
talk. So there are problems, and they talked about this reliability ever droplet movement, and also the sample preparation based on that. Okay, so basically it changes basically the volume, and also it will cause the means effect. It will affect the accuracy in the protocol. Yeah. Yes.
Thank you, sir.
Yeah, thank you.
Thank you once again, Dr. Shudip Roy. Now, I would like to request our HOD sir, Professor Shoykot Maiti, to please felicitate our speaker. Sir.
Am I audible?
Yes, yes.
Yes, sir, you
Sir,
are.
thank you so much for this wonderful session that you have given to our faculty members as well as the many faculty members, those who are present in this FDP. It was really a nice session. I have heard this session from the beginning, and it is very interesting indeed. Please accept our regards, love, and token of appreciation from School of Engineering and Technology, JIS University. Please accept
Thank you.
this, sir.
Yes, sure. Thank you. Thank
And
you for your kind words.
thank you, sir. And a small digital e-memento for you, sir.
Thank you. Thank you
Thank you
very
so
much.
much. Thank you. Thank you, sir.
Thank you very much, sir, for this wonderful talk. Thank you so much.
Thank you very much. Yeah. Thank you. Okay,
So
so I
over
can.
to Dr. Datta.
Oh, okay.
So probably we'll start the next session from 12 o'clock.
Okay, bye
Thank
for
you.
now. Yeah, bye.
Okay, bye.
Bye, sir.
So Dr. Banerjee, we will start the next session from 12.
Definitely, definitely.
Thank you so much, participants, but please be here. That will be a wonderful session to relax all of us. So please be here.